Our next presenter this morning is Andrew Kolmatiski.
Yep.
It's almost as challenging as Demarest. Andrew is a research professor in the Plant, Soils, and Climate Department at Utah State University. He's been exploring the role of microbes in water use by native plants and weeds in the Intermountain West for the past 15 years. And this morning, Andrew's going to talk a little bit about how soil-root interactions can be used to manage plant communities. So please welcome Andrew.
Thank you. My wife has told me that I drink too much coffee before my talks and that I make people's heads spin. I say I'm talking about soil, so I'm supposed to make people's heads spin. The idea is to give you a new worldview on how your systems are working. And she says, no, it's because you drink too much coffee and you talk too fast. But you're in luck because I'm stupid and I had a grant due yesterday. And I thought I had a 15-minute talk. But I have 45 minutes. So I can take it nice and slow, walk everybody through. If you have any questions, just jump right in as I'm going along here. Let's see. I think this talk thing might be preventing me from doing anything. Okay, here we have. So I'm going to try to introduce you to a new world. This is a crazy place. It requires a whole new perspective on how the world works. This is an area where species concepts degrades. There aren't really species. For bacteria and fungi, we talk about operational taxonomic units. As an example, this crazy place. Has anybody seen these glowing mushrooms? Pretty cool to find. Here's a bacteria that is transferring genes within its life. It'll transfer plasmid DNA to another bacteria. So a bacteria within its lifespan of hours has new genes in it. And so its genetic material is different within its lifespan. Okay, and these things are reproducing hundreds of times a day, so evolution is extremely rapid. And so does anybody know what this is? This is a plant root, but it becomes very morphologically different when it's covered by ectomycorrhizae, right? So you have these associations between fungi among different kingdoms where fungi and bacteria will grow into plants, change their morphology and physiology. These are root nodules where plants create these abiotic environments for rhizobium to grow within their roots. And so you have these mixes, these hybrids of organisms from different kingdoms that really determine how plants grow, die, compete with others. You even have situations where trees eat animals. So this is a root-trapping fungus that will form associations with plants. The root-trapping fungus has these trigger hairs. The nematode swims through. The fungus traps it, digests it, and then that fungus sometimes is connected to trees and plants. And so the plants get the nitrogen from the nematodes. So you've got these carnivorous maple trees do this, for example. And how does all this happen? It's all the underground communication superhighway. So this is all by what's called blebbing, very technical term, where bacteria, for example, will pop off a little bit of their membrane with some communication signals in them. Bacteria talk to plants, plants talk to bacteria. This is how the rhizobium knows to find the root of a certain plant species. It's now how a pathogenic fungus knows to find the plant that it's going to consume. And the past few years there's been some amazing research showing how, for example, bacteria will release auxins to increase the growth of certain plant roots. And it'll be species specific. Or plants will release anti-quorum sensing chemicals. So bacteria will grow almost socially. They release these chemicals called quorum sensing chemicals. And the more of them that are in the soil, the faster the community grows or the slower the community grows. And plants have figured this out, and they release these chemicals to slow the bacteria down if they don't want them or encourage the growth of others. So the bottom line is there's all this communication going on among kingdoms, plants to plants, plants to bacteria, bacteria to fungi, fungi to archaea. And this is important to remember because this is where, as a rule of thumb, half of plant life exists, right? So I love this picture. I don't think that's an Oompa Loompa. I think he's an almost regular-sized person. Nice grass root mat here. You can get spectacular growth. I've got cheatgrass pots that I've planted in deep pots. In 2 months, will completely fill the pot with a root mat, right? So a lot of the energy that plants fix from the atmosphere is going below ground. Okay. And again, if that's confusing, jump in anytime. We have lots of time here. So I'm going to talk about 14 is the magic number of the day. I started working near Winthrop, Washington on the dry side about 14 years ago, and starting my PhD research. And one of the things that really struck me were these fence line distributions. And this is something I'm sure you've all seen, something we've seen around the West. This is a 50-year-old, it's now 60, it's 1955. This was abandoned from dryland wheat production. And for the past 50 years, it's been dominated by weedy plants, annuals, mostly annuals and biennials. This is all diffuse snapweed. And the thing I love about this picture is this is a ratty old fence. It's not really keeping anything out. And this is a pretty healthy blue bunch wheatgrass, pseudonaria community. There's a lot of balsam root and lupin right around here. And it's downhill and downwind of this knapweed field. I mean, here you've got, this is like, what, 10, 15 centimeters here, healthy native bunch grass, weeds for 50 years, right? And so for the past 15 years, my research has essentially been trying to explain this. What is going on here? Why do we have this weedy community here and this native community here, and why don't they mix? How come there's no succession? What's going on? What's explaining this? And the long-term goal of this research is, if we can understand what's keeping the weeds where they are and the natives where they are, maybe we can find out, if we understand those mechanisms, maybe we can apply them to direct population growth or native plant restoration or weed control. So one explanation is that you've got these non-native plants and they're just superior competitors due to differences in coevolution or because they left their enemies behind. Maybe they're just better growers. The problem with that is that you'd expect, if they're just superior plants, you'd expect them to be invading over time. In a lot of areas, you see these native communities are pretty resistant. This has been for 50 years in this case. Another good explanation is that there's some legacy of agricultural use. So the soils have been tilled, the native fungal networks have been ripped up, nutrients have been, fertilizer may have been added. In this dryland case, is not too much. But the soil may be looser. There may be legacies of this agriculture, and maybe that's still driving the abundance of these plants. Alternatively, maybe they've escaped their enemies, but again, this isn't a very satisfying explanation because then you'd expect them to be superior growers that would eventually invade the native communities. Could be, herbivory. Here's a grazed hillside and an ungrazed hillside. But again, this isn't a very satisfying explanation. There's a lot of mule deer here, and this fence isn't keeping anything out. People suggested climate change, but again, with climate change, you'd expect, that if this is just now a better environment for these species, you'd expect them to gradually be invading. Now, the alternative, as I was starting this research, plant soil feedback research, was really just developing, and the concept here was that a plant, whenever it grows, will accumulate pathogens or symbionts. It changes its soil environment in a way that can either increase or decrease its own growth. So maybe once these invasive species get established, they are becoming ecosystem engineers, and they're creating a soil environment or an environment that benefits their own growth. And so I wanted to explore that with my research. Give you a little bit of a background on what are plant soil feedbacks. The general idea, as a plant grows, it accumulates nematode pathogens, for example. Those nematode pathogen populations increase, and that decreases the growth. So that's the feedback. But it's not just pathogens, of course. There's also symbionts, mycorrhizae, growth-promoting bacteria that could increase plant growth. And of course, it's not just one plant that we're interested in. You could have a grass that increases the population of this nematode, this pathogen, decreases its growth. But it's possible, and there's cases where it's been found that a plant increases the abundance of a pathogen that decreases the growth of its competitors even more than it decreases its own growth. So it's really important, though rarely done, to consider these community-level plant soil feedbacks. And just more generally, how do plants change the organisms in the soil, and then how does that affect the whole plant community. Is that making sense? Okay, so just as a little example from some of my research to put a face on it, this is a plant soil feedback study that I'm running up in Washington with 1,500 quadrats. And since we have time, I'll tell this little story that I like about this. And when I put this, I was young and ambitious. I put this experiment together, and I was like, oh, I could do this treatment too. So I started out with like a 200 plot experiment, went up to 1,550 plots, and I had to do it by myself because I had no money, and I was doing my own research. So I had to put all this down, and I put like 4 tacks in for the felts, the whole 1,500, and then a tag in each one. And so for each plot, I had to put these things down. So I had a backpack full of pins and lunched down, put the pins in like 6,000 times. Then I went for a bike ride after doing this for 2 or 3 days. My bike shorts were tight on this leg and loose on this leg because my leg had grown about 1.5 inches from doing about 6,000 squats in 2 days. But after a few years of research in this, one of the species, this is a feedback experiment, so we can plant, like for example, monocultures of certain species, let them grow for a few years, and I killed them all, and then replanted to see how plants in the next stage grow. And one example, I planted Colomia grandifloria. This is a native, a pretty common native, but it's never really abundant. It's kind of all over the place, but never really abundant. And I had seen it. It usually grows between, like, there's a few different species, but it grows like this tall to this tall, and then it'll seed, flower and seed. And I planted this in this year, in the first year of this study, and I had on average 50% ground cover of Colomia, and they were generally above my knee. And when I first went to the plots, I was like, what is growing here? Because I didn't recognize it. It had just exploded. I'd never seen it grow like that. And then it flowered, and I was like, that definitely is Colomia. And then the second year of the study, I seeded again. There was all the native seeds there, and I had 0 growth. There was not a Colomia plant to be found. But this is what I found. There was a fungus that developed in all those plots where the Colomia had exploded, and it just hammered them. I mean, it just totally wiped them out. So there's that feedback effect, right? The plant grows in place, and then it accumulates those pathogens. Interesting, we're seeing these damp cycles. The third year, that fungal population had nothing to eat because it killed everything, and then the Colomia started to come back. The fourth year, we see it drop again. So these seem to be not only conceptually important, but here's a real-life example. I did a meta-analysis a few years ago, so a review of all the literature. There's different ways to measure plant soil feedbacks, but I compared across the literature. These 2 red bars are different ways of measuring plant soil feedbacks. But across the literature, what effect did they have on plant growth? And so when you compare them to other meta-analyses of seed feeders, adding leaf litter, adding pathogenic fungi, adding above-ground herbivores, doing soil warming, adding root feeders, herbivores, it seems to have a pretty big effect on plant growth on all papers across the literature. It seems to be about as important as herbivory. So literature is suggesting that it's a potentially important effect. So I went to these sites, and I had seen this pattern. We've all seen it, but I wanted to do a little more controlled study just to determine really that these weedy fields are really weedy and stay weedy. I was only there for a year or 2. I wanted to confirm that this pattern is real. So I went to 25 abandoned agricultural fields around this valley and found out what year they were abandoned, and then just surveyed 5 meters into each field and then 50 meters into each field and looked at the plants present and confirmed what seemed to be the case. So if you have exotic vegetation in yellow at the tillage boundary, when I went to all these 25 randomly selected fields, weeds dominated regardless of how old the field was. Fields could be abandoned in 2010, it could be abandoned in 1955. It was dominated by annual and biennial weedy plants. And there didn't seem to be much migration. The native plants don't seem to be moving into the fields, and the weedy plants don't seem to be moving out of the fields. Another way to look at this is just a description, multivariate statistical way to describe the whole plant community. And so here, this is described by functional group. Here's the native communities. at 5 and 50 meters, and here's by species composition, and then here's the weedy communities. And what this really showed me was that these weedy communities, you can see, are all over the place. These lines connect 3 points, the species composition over 3 different years. And so these weedy communities are changing a lot each year. One year it's knapweed, the next year it's cheatgrass, the next year it's whitetop. But it's always going exotic, 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 all over the place, different species, different functional groups, exotic grass, native, or exotic forb. And the native communities, as you'd expect, 50, 100-year-old bunch grass isn't changing. It's sitting there in the native community. So this kind of also is some support for the idea that these weedy communities, they're very dynamic, and it's consistent with there being feedbacks in the soil. Yeah.
Is that because of the different climate during that year?
So, it could be, it's going to be a combination of, the climate that year and the previous year probably, how much seed set that there was from other communities.
Cold in February, hot in March.
Yeah.
Mustard, something else.
It's it would be hard to get. I now have this survey data set for 14 years now, and I'm just about to start analyzing it for climate signatures across the 14 years. But the alternative is that, like we saw with Colombia, you have a big year, it has a lot of fungi. So you could get knapweed might have a really good year, and then it might get hammered by a Larinus or get hammered by a soil fungus, and then whitetop comes in the next year.
Another question.
Yeah.
How much organic matter do you have to have in the soil to get a good wet soil base?
To get a good what? So this?
The bacteria.
Oh, they're always everywhere. I mean, if you have a lot of, there's also a trade-off between having a large microbial mass and a large microbial activity. It's really the activity. And so one thing I've found with the weedy communities is they actually create small microbial communities, but they're very active. And so the pools are small, but the fluxes are large. And so just looking at the size of the microbial community is probably not that important. Also, most of the microbial species are just heterotrophs that don't have much to do with plant growth. So I did another study in this system where I planted, so I cut out plots and then planted either weeds in weedy fields or weeds in native fields, where I killed the plants that were standing, natives and weedy fields, to try to get an idea of the soil history. How important is this soil history? Is there something inherent to those soils that's creating the distribution that we see? And it was really the overriding effect. So we see this huge, and I'm focusing on exotic plant cover here because the natives pretty much didn't care. They always had poor growth, poor cover. No matter what treatment I applied, they would always do about 6% ground cover. Didn't matter where it was. The exotics were had big differences among treatments, and they really loved their own soil. They did really well in formerly weedy soils. I compare that to competition, for example, so whether I left the plants standing or not, sure, that suppresses weedy growth. Not that much. It didn't really matter to them. The really important thing was that soil. They did really well on weedy soil. They didn't do very well on the native soil. So it suggests either that the weeds are creating a soil that encourages their own growth, or that the native plants have this bacterial or microbial community that suppresses the weed growth. Compare that to tillage. Tillage didn't have much effect. Adding a fungicide, when I added a fungicide to these weedy fields, that's kind of pretty strong. It was the next strongest effect. And it did a pretty good job of suppressing the weed growth, and so it seemed like there was a mycorrhizal or a fungal symbiont accumulation that encouraged weed growth in those weedy fields. So that's kind of the state of the art. Most plant soil feedback research just sort of measures, is there a difference? Do plants grow differently on different soils? And that's kind of most of what people have done. My research in the past few years is focused on what can we do with that? Can we really use that to predict how a community will develop? And can we use that to then say, well, you should put these 4 species together, or these 6 species, or these 10, because they're going to grow well together, and they're going to be really vigorous. If you put these 5 together, this one's going to have a pathogen that will kill these 3, and then you're only up 2 native species. So, and that'll leave a lot of niche space open for the weeds. So can we really use this plant soil feedback data to predict plant community composition? So this is a study where I just do a classic plant soil feedback study. So you grow a monoculture, then you replant. And so this is an example can't tell from here. I think that's cheatgrass. Cheatgrass growing, I think it's growing on self soil, and then this is on, this might have been Agrippine cristatum. I think it really didn't like Agrippine cristatum soil. But you can see there's a difference. Depending on which soil you grow it on, plants will grow differently. And then that's all I take. What's the mass of a plant species on its own soil versus the plant mass that it attains in soils cultivated by other plant species. And then I run that through a plant cell feedback model that I've developed and predict plant community growth. And then I plant, and this is in Tom's labs, thanks to ARS for lending in the greenhouse space. And then I take pots and I put those species together and see how well they do. And what I find is that, so this is for blue bunch wheatgrass, Galeric strata, and Hesperostypa camata. So here's a competition model. So if you just based it on how would this community look based on plant mass, Calaria in this greenhouse experiment did the best. So a competition model would predict that Calaria would be most abundant, then Stipa was next, and then blue bunch in this case was the worst grower. And so that would be the prediction of a competition model. When I add these plant cell feedback effects in, how does blue bunch grow on Calaria soil and how does Calaria grow on blue bunch soil? It shifts the order. And so it actually got the rank order correct. It predicted that blue bunch would be most successful, Calaria next, and Stipa camata would be next. And that's what we observe in these white bars. And so for all the communities that we examined, just measuring these plant cell feedbacks improved our ability to predict which species would coexist, how successful they'd be. So it seems like these things plant cell feedbacks are important at sort of on the landscape scale, and they seem to be able to help us predict how a community will come together. So the idea is we may be able to use this information over time to construct plant communities or seeding plans that will be more successful. This is just another test with it. That was one example for one community, but when we do it with a number of communities, we do a pretty good job. So this is a prediction of over-yielding. This is for sort of a slightly different experiment, but predicting plant mass and how well species do. And for ecology, R squared of 0.2, I'm happy with it. I'm predicting, this is R squared for my ability to predict the exact number of grams of a plant in a community. So what can we do with this? Is there some way we can use this information that the different plants create different microbial communities to restore our landscape? So again, this all occurs as a result of communication among organisms in the soil, right? And in general, what we see is that across all feedback studies that have been performed, plants accumulate pathogens. Pathogens they evolve very quickly, and they know how to hammer plants. They know how to eat them, right? They're really good at it. That's all they do. They evolve quickly to do it. So in general, plant cell feedbacks are negative. When you grow a plant, remove it and plant it again, then the next generation is going to grow worse because it accumulates pathogens. Weeds don't have that effect as strongly. They tend to have less negative or even sometimes positive plant cell feedbacks. They'll actually grow better in the next generation. So this all occurs because a bacteria knows that a plant is there. And it knows it because the plant root releases these small organic compounds. So it's this communication by these small organic compounds. So we thought, well, what if we can shut that communication off? We should be able to remove the benefit realized by weeds and remove the detriment caused by pathogens to native plants. And the way we thought to do that was to add activated carbon. It's like a molecular sponge. Just through van der Waals forces, it'll hold any organic molecule. Okay, it's also used in your water filters. It just binds anything. It's just like a sponge. So the idea is, if non-natives have these positive feedbacks, alternatively, non-natives have been suggested to be allelopathic, and that's also a chemical signal that moves through the soil, and that may decrease native growth. And non-natives may also increase nutrient cycling by their effects on soil bacteria in particular, and that can increase their growth. If we could add activated carbon, we can shut down all those mechanisms, and that could potentially lead to an increase in native relative to non-native growth. So we gave it a try, and we had some pretty remarkable success. I couldn't believe it. So this picture is a little dim here. But so here is a plot where I put native seed down and got absolutely no native growth. And then you can see these plots, these quadrats in 2 transects here, where I added activated carbon and native plant growth. So these are each activated carbon treated plots, and there's like a control seeded plot in here.
So you do carbon and seed.
No more questions. This is a secret part of the trip. Yeah, I added native seed. Yeah, there are plots in here that have activated carbon and no native seed, and there is a native seed limitation. This whole field has been abandoned. So that whole field's been abandoned for 55 years, and natives haven't gotten in. And yeah, so you have to add the seed, definitely. But we have remarkable success. This picture, I think, is 6 or 7 years after a single application. So I went in, added activated carbon once, mixed it in the top 10, 15 centimeters of soil. Natives loved it. So this is Blue Bunch in particular loved it. So given that initial success, we got some funding from USDA to take our show on the road. And instead of doing a few pounds, I bought 20,000 pounds of activated carbon. That's just that's just going to one field there. Anything you do with soils is heavy and big. So we did a set of 15 by 15 meter treatments, and we added this at 1 kilo per meter squared, super high doses. We just wanted to see if it would work. It's really expensive, prohibitively expensive to do it like that. So we wanted to test a number of concentrations to see if we could get the prices down. So we did 100, 400, 700, and 1,000 grams per meter squared in these large plots. It's dirty work. I have a graduate student now who's found a cleaner way to do it. But so these are just some of our 15 by 15 plots. And here's a picture after, I think this is 1 or 2 years of growth. So here we have our different concentrations and then some control plots. This is all white top, and you can kind of see, it's a little hard to see, this is mostly native grass that you see growing in these carbon-treated plots. We also tried a wood-based activated carbon. And here's the results. So control, this is just native to invasive ratio, so how abundant were the natives? Very low, one quarter native growth, and that's mostly native annuals. Seeding really helped in this treatment. Put these treatments down when they happen to have a really wet year, so we actually had really good native germination this year, which actually kind of reduced, I think, our treatment effects. But unfortunately, we found 100, 400, 700 didn't. We see a similar effect. We get native plant dominance with this 1,000 grams per meter squared. So at high concentrations, it seems to really boost up the native plant growth. But these lower concentrations don't seem to be effective. So it seems to be a costly approach. I'm now working actually with, just started a project, Paul Grossel at Utah State. One potential application, even when it's this expensive, is this is a Google Earth picture of Price, Utah. These are all oil pad exploration sites. And so it'll cost a couple $1,000 to put down carbon on these basically 1 acre plots, but it's something that I think they'll be interested in to get native plants growing, so we're testing that there. But we're talking at $10,000 a hectare at 1,000 grams. So for the most part, cost prohibitive, and so I'll talk now about some of the research that we're doing to try to find some ways to be more cost effective. So how does it work? I mentioned a lot of native plants change nutrient cycling regimes. So cheatgrass I found and the weeds in this site, for example, increase nutrient cycling rates, so increase net nitrogen mineralization. So does activated carbon change that? Well, we found actually, if anything, we found non-significant results, but if anything, AC seems to be increasing nutrient turnover rates, so that's not a mechanism that would encourage late successional native plant growth. It would encourage weedy plant growth, so it doesn't seem that activated carbon is changing plant growth by changing nutrient availability. Allelopathy is an option, and so what we've done to test this is I did a greenhouse experiment where we've added activated carbon in live and sterile soils. So if activated carbon works in sterile soils, then that means that it must be having an effect on allelopathy, some direct root exudate that directly affects another plant. And in general, I can show you some species data later if you're interested, but the general pattern is that activated carbon only seems to work in live soils. When you add it to sterile soils, it doesn't seem to have the effect. There's a few species pairs, and there was even some native species that were allelopathic on weeds. But the general effect is that it seems to be microbially mediated effect. We can also use this to, I've looked at the microbial, exactly how does activated carbon change the microbial community composition. And one interesting thing is this is using phospholipids. So here's a native the microbial community under native soils, so associated with native plants. And then here's how weedy plants change it. They have a huge effect on the composition of the microbial community. In general, they suppress total microbial activity and abundance, but they do increase the abundance of a few particular, in this case, lipids. And then when we add activated carbon also reduces the microbial community, but it particularly reduces a few of the lipids associated with weedy plants. So this is pretty strong support for the idea that activated carbon is doing something to basically remove the benefit that weedy plants create in their own environment. And I've repeated this recently using a much more detailed pyrosequencing technique, and we're actually seeing a pretty similar pattern where when you add you have native community, go to weedy community, add activated carbon, and you start to return that microbial community. It starts to look more like the native community. And I'm going to come back to that a little bit in a minute. Yeah.
Have you studied, compared, or with the
I can repeat the question too.
Terra, how do you say it?
Oh yeah, yeah. So
would that be more
cost effective?
cost-effective?
Yeah. So there is, and I haven't linked up too much with the. So the question is activated carbon similar to the charcoal treatments or terra preta? And basically, the charcoal treatments should be similar. Activated carbon is basically cleaned up charcoal. And so it's charcoal that has where a lot of the junk in it has been removed, so it's only a carbon skeleton. And so it's basically much more effective charcoal. So I think it's a little more costly to buy activated carbon, but it's going to be much more effective. Okay.
If we could leave the questions then and talk, that would help us in the recording.
Okay, sorry. Okay, so the activated carbon treatments, we see that it's changing the microbial community composition. And in a separate line of work, I have this plant soil feedback experiment. And so I can look at how each species that I've planted changes. It's a common garden experiment. I'll start with the same soils. So by growing each of these plants over time, how do they change the microbial community in the soil? And when I do that, you can see I haven't pulled out a consistent pattern with native versus exotic. So here's blue bunch, balsam roots. Here's tumble mustard right in there, pretty similar with them. That's lactuca, knapweed. Here's colaria. So there's not a real obvious native, exotic, or foregrass grouping, but the species are all pretty unique. Especially cheatgrass really stands out. Tragopogon really stands out. But they're all create very distinct microbial communities. And one thing that I'm doing now, and a really nice thing about having this experiment and the activated carbon treatments, is that I can now cross-reference the microbial species and there's thousands of microbial species in the soil, so I can now cross-reference those species that are identified or created by a particular species with those species that activated carbon affects and a reduce in growth of particular weeds that activated carbon causes. So by cross-referencing these lists, I can come up with a list of microbial species that are most likely to suppress weed growth or most likely to encourage a target native plant growth. And so this research I've just been starting to analyze this data in the past month, but that's sort of the direction we're going. And so the holy grail here's the first 10 species that I've pulled out as most likely candidates for, and we've just started with the bacterial analyses, the fungal analyses are still being performed. But the species that jump out for potentially increasing blue bunch and lupin growth and bacterial species that are likely to decrease cheatgrass, Tragopogon, knapweed, Cicimbrium locellii. So I can now reduce this list of thousands of bacterial species down to 10, 20 that I can really target, that we can really focus on to try to identify biological control agents. And the idea is to hopefully be able to cultivate these bacterial species and use them as inoculations to control native plant growth. As an example, and I'm curious, it's nice the following talk I think is going to be on this topic, but so I'm not sure where this research is at, but I've been fascinated with the fingers of death research, which seems to be a native bacterial pathogen for cheatgrass. And I'm not sure, I'm curious to find out how, I don't know how they found this species, but basically my research is a way of taking lists of thousands of potential species and narrowing it down to a few that we can really focus in on as potential agents. Not sure how much time I have left here, but perfect. Two other sort of sidelines in this research. Plant cell feedback research sort of typically focuses